So I will try to go quickly through the main conceptual idea that we are dealing with now at the moment. So basically, first, how we relate, the main idea is how we try to relate brain organization to a range of behavioral function with activation data, and then what does that mean in terms of how brain process information. So first of all, how do we study brain organization when we are interested in large-scale in integration across a large-scale network? We use connectivity-based parcellation. So really the basic idea is that if we are interested, for example, here in the dorsal premotor cortex, we take the voxel within this region, we compute the connectivity with all of the voxel in the brain. We put this information in a connectivity matrix, and then we're going to use a clustering or a factorization approach to somehow summarize, represent what is in the data. So what the clustering will do is to look basically at the similarity of the voxel and enforce a subdivision into different group of voxels that show similarity in their connectivity profile. So here, for example, we enforce a parcellation into 5 clusters, and this is what best represented the data here. And what we see is an organization of the right dorsal premotor cortex along 2 axes, one which is rostrocaudal and one which is ventrodorsal. As I said, this is the clustering that best summarize or represent the data, but we could have also subdivided this region into 3 clusters, and then we would have only a rostrocaudal organization with a rostral region, an intermediate region, and I can show it here, a rostral region, an intermediate region, and a caudal one. I can show you a different example in the hippocampus in which we have also different dimension of organization. So if we use functional connectivity in terms of coactivation and resting state functional connectivity, what the clustering algorithm would do is first subdivide the hippocampus along the longitudinal axis. So it will differentiate anterior to posterior region. And this pattern that we get, that the clustering highlight in the data, actually converge with other approach from some collaborators in Montreal. They use a gradient approach, and what they show using this, it's a kind of PCA, so they extract the first dimension that best represent the variability in the connectivity profile. And what they show is that they could find a first gradient, a first dimension that they characterize along the longitudinal axis in different subregions of the hippocampus. So this really fits with how a clustering algorithm was identified here, this main first dimension that drives the differentiation in functional connectivity in the human hippocampus. But then if we keep on splitting the hippocampus voxel, what we see is medial lateral differentiation that mostly appear in anterior region. And if we go further and go to higher level of granularity, this becomes really obvious that we have this medial lateral differentiation. And this fits with the second gradient that our collaborators in Montreal have highlighted in the hippocampus subregion, which they characterize along the medial lateral axis. And interestingly, this medial lateral differentiation also fit well with what we know from cytoarchitecture. So studies that have looked directly at the microstructure of the hippocampus have highlighted a subdivision into different subfields that are more or less along the medial lateral axis. I'm simplifying when I'm saying that they are arranged along the medial lateral axis, but this is just for getting the main conceptual idea of our work. And interestingly, if we use another parcellation modality now based on coplasticity, so how gray matter volume will vary between brain regions across a group of subjects, and if we use this information for looking at the organization of the hippocampus with a clustering algorithm, the first thing that we see is that the clustering first differentiates along between anterior and posterior regions, so along the longitudinal axis, and then already at a three cluster solution level we have a differentiation along the medial lateral axis, which really resemble what we see from cytoarchitecture as subdivision between subiculum and CA field here. So in other words, to summarize, I hope you can see here that we cannot come up with one map of the human hippocampus organization, but we have to consider that there are different organizational dimensions, and cytoarchitecture microstructure mainly primarily capture this medial lateral organization, and resting state functional connectivity reflecting large-scale functional integration will primarily capture a differentiation organization along the longitudinal axis, differentiating anterior and posterior region. Then the next question is, if those gradient and organizational dimensions are true neurobiological feature, what do they mean? What do they reflect in terms of how a brain processes cognitive information? To address this question, we can link the data to behavioral function using activation database, and the most famous one are BrainMap and Neurosyn. So Neurosyn has been developed by Tal here, and they have been developed completely differently, but they both have similar information in the sense of for each study, each activation experiment which has been collected in the database, they have information about where activation has been reported in the brain and what type of behavioral condition was used, what type of psychological concept were investigated in the particular study. It means that for any brain location or any brain region, we can probe those databases and identify the most frequently associated behavioral function or psychological concept. If we do that in the right dorsal premotor cortex parcellation that I show you, we see, for example, that the dorsal cluster is associated with finger tapping, the ventral cluster is associated with function probing, more eye field function. Well, this is not very informative when taken in isolation, but what is more give more insight is to look how the behavioral profile change as we travel across the organizational dimension. And what we see if we characterize the rostral-caudal dimension is we really see a shift from higher cognitive function in the rostral part toward more sensory motor paradigm in the most caudal part. So, of course, this type of hierarchical processing, in particular in the frontal lobe, was already known from previous studies. So here this is more like a proof of concept. And in the human hippocampus, there have been theories before to explain this anterior-posterior organization in terms of encoding versus retrieval processes. But this is not what we see when we look at association with behavioral function in Neurosyn. So we see activation for both type of function all along the longitudinal axis. In turn, what other studies have suggested is that there would be a shift from the anterior region to the posterior region between emotion to cognition, and this is indeed something that we see, this emotion-cognition gradient in BrainMap and Neurosyn. However, we also see that there are associations with cognitive function in the anterior part. So another hypothesis that we came up with when looking at the data with Neurosyn is that there would be a shift from anterior to posterior region, maybe in terms of self versus word-centric mode of processing, self versus word-centric type of representation. That would be important for functions such as spatial navigation, but also memory, episodic memory, and maybe also language function. Okay, so to So we have to note that all these results are based on aggregation of a lot of results from group studies, group experiments. So they are, of course, a very limited spatial precision, but there is also a publication bias because all these activation studies, they are only studies that have been published in the scientific literature. So we only have access to this type of data. However, what would be interesting then is to have subjects scan across a very wide range of paradigm, and this is now what individual brain charting projects offer. So this is fully open access, and this has been developed by Bertrand. So this opens new perspective to linking this organizational dimension to the range of behavioral concept, behavioral function that we have in psychology and other fields. Okay, to sum up, I hope I could show you how behavioral profiling of parcellation map could help us to understand the relationship between aspects of brain organization in terms of gradient and dimension to behavioral function, and then to infer about what it means in terms of how a brain processes information, and the new perspective with individual brain charting data that we have. Before I conclude, or before I give the mic to Tal, I would like to just emphasize that I'm really happy to be at this symposium with great speakers, both men and women. I know that it's difficult for organizers to find women. It's not always easy. Women are not visible, so to help in that regard, we have created the repository for women in neuroscience. We have now more than 1,000 profiles. So if you are a woman in neuroscience, please register. If you are a conference organizer, please use our tool to find the right profile of a woman for your conference or workshop. And whoever you are, please submit recommendation. There are a lot of women giving talk at this OHBM meeting, and a lot of them have a profile on the repository, so if you want to support them, just share your positive feedback after the presentation by writing recommendations. With that, I would like to thank you very much for your attention, and to thank my collaborator, my research group in Jülich Research Center, as well as my collaborator in Düsseldorf and beyond. Thank you very much.